around, and thanks to the organizers for inviting us. Today I'll be talking about our project looking at regulatory variation in the 3D genome. So regulatory variation is DNA sequence variation that occurs in genomic regions that can influence gene expression. And so only about 5% of the GWAS SNPs in the NHGRI-EBI database data catalog are in coding regions, and 93% of them are non-coding regions, with some of them located 100 kilobases to 1 megabase away from anything that's coding. And so it's really important to try to understand what these non-coding variants are doing, how they're influencing the trait, what pathways they're targeting, so on and so on. And so one of the ways in which GWAS variant can act on a gene over long distances is through long-range regulation. And so that's when a distal regulatory element, such as an enhancer, loops over and comes in close three-dimensional proximity to the target gene. And from there, it can influence gene expression, but there could be a variant in that enhancer that could affect transcription factor binding, for example, and that could influence its ability to affect that gene expression. And as you may know, the 3D genome has been implicated in many diseases, and it's also important for development. So here we have deletion of this TAD boundary would cause these limb-associated enhancers to start to interact with genes that it normally wouldn't interact with, and that could cause things like F syndrome, and polydactyly is also caused by a similar deletion. And so it's also important, we see changes throughout mammalian development as well. And so there are a number of different experimental approaches to detect long-range interactions. I'm going to be focusing today on HIC since it has genome-wide coverage. But as was already alluded to, high-resolution HIC data is rather limited. And so here, for example, we have two real loops. And if you did a HIC experiment, you would get paired reads, which you would theoretically be able to map enhancer 2 to gene 1, for example, and enhancer 1 to gene 2. But as a processing step, we tend to aggregate the signal into bins. And this is because the sequencing depth from HIC experiments is pretty low. And so you're really saying bin 1 interacts with bin 3, but then you lose those two loops. So it doesn't really recapitulate the loops that are present. If you had a better sequencing depth, you could have a higher resolution and a smaller bin size, and that would make it easier to tease out these interactions. So here, if you went down to a 5 KB bin size, you would be able to tease out these two real loops. But HIC data at this resolution is relatively limited. There is some out there, but there's not much. What we do know is we do know that looping is associated with various chromatin states and architectural proteins. So here we have the SOX11 promoter, which is clearly marked by different peaks for the epigenomic signals and the architectural proteins, and this is associated with limb development in mice. And the red and the green elements here are things that it's interacting with, and you can see that they are clearly marked by peaks as well. So we know that there's this association. And while there's not a lot of HIC data that's out there, we do have a large amount of epigenomic data thanks to large consortia, such as the Roadmap Epigenomics Project, which measures epigenomic states in various human tissues and cell lines, as well as data being generated by individual labs. So there's really a lot out there. And so we could leverage this data to generate predictions. And so that's what a graduate student in the lab, Shilu Zhang, did. She essentially used these features, fed them into a model that is trained on the high resolution HIC data that we do have in order to generate the predictions completely in silico. And so we validated this in a number of different ways. We looked at the CTCF, on the looping level, looked at the CTCF bidirectional enrichment. So that plus minus orientation that indicates a presence of the loop in the predictions. We also compared it to CHIA-PET datasets as kind of gold standard to see if the predictions were enriched for those. And on the TAD level, we used TAD colors on the true counts as well as the predicted counts, and saw that they recapitulated the TADs that were present in the true data. And so we took data from the Roadmap Epigenomics project and used these models to predict high-C data in 55 cell lines so far. And so what I'm going to be focusing on today is getting the significant interactions from these 55 cell lines, looking at the SNPs that are in the NHGRI-GWAS database, and then mapping those SNPs to genes. One of the problems that we ran into pretty early on was that while there are a lot of cell lines in the Roadmap database, they don't all have the same marks available. And so when you're training models and you need a specific set of features, this could get pretty annoying, as you can imagine. And so even if you took the intersection of all the marks that are commonly measured in the Roadmap database, you'd only end up with 22 cell lines. So if you wanted to train on those specific features, and that's where the Noble group came in and really helped us out. They have a method called Avocado that's for literally doing this. So it basically imputes the data set as long as you have data for that cell line and for other cell lines as well. So that brought us up to 22 to 55 cell lines. And so there are 23,000 non-coding SNPs in the NHGRI-EBI GWAS catalog that we considered. And so just using the predictions to map them to genes. Again, we have 55 cell lines. This is the number of SNP gene interactions. So there's about 15,000 SNP gene interactions per cell line. And this is regardless of phenotype. So this is just a general overview. And so I already talked about how we could use the HIC-Reg predictions to map the SNPs to target genes. And that would tell us what the target genes are. But that's only part of the picture. We really want to get a really complete picture of what these SNPs are doing. And so we have the HIC-Reg predictions again. So we have the SNPs mapped to genes via long-range regulation. And then we can take molecular networks. So we use networks that are derived from transcription factor binding and protein-protein interaction networks. And then so essentially what we want to do is try to use this network structure to try to get at the pathways that are present. So essentially we score the nodes that are there based on their significant interactions with SNPs via long-range regulation. So if you're a SNP that is, if you're a gene that is interacting with SNPs related to a particular phenotype, then you would get a higher score. And then to try to get more at the pathways that are there, we did node diffusion. So that'll tell us what the neighbors of those genes are. And so what we want to come up with is a way to get really network-aware modules from this. And so the way to go about doing that, or at least our approach, was we did edge diffusion first. And so this would give us an edge weight adjacency matrix. And then on that, we did graph clustering. So essentially, this would give us the different components, which could hint at different pathways that these long-range interactions are targeting. But the question now is, if you did this, if you did graph clustering on 55 cell lines individually, cluster 1 in cell type or tissue A doesn't really correspond to cluster 1 in cell type B. So we really wanted a systematic way to compare the different pathways across the 55 cell lines. So the answer to that, at least, is to do multitask graph clustering. And this would give us more of a systematic way to compare across the different cell lines. So essentially, we start out with a data similarity relationship. So in our case, we use H3K4ME3 and the promoters of genes, and then did a hierarchical clustering for the 55 cell lines. And then, as I mentioned, we have an edge weight adjacency matrix for each of the 55 cell lines. And so we'll do multitask graph clustering or multitask spectral clustering. And so this is spectral clustering, but it's going to be constrained by the data similarity relationship. And so more similar cell lines will end up having a more similar graph clustering. And so this gives us an automatic mapping of the clusters across the different tissues. And so we can compare the cluster 1 from tissue 1 to tissue N. So the green cluster and the red cluster shown here are pointing to the same clusters across the different tissues. And so now I'm just going to talk about one of the phenotypes we looked at, resting heart rate. So there's 33 GWAS SNPs here, lots of LD SNPs, and this is just going over the number of SNPs, the number of SNPs, the number of genes, and the number of SNP gene interactions across the 55 cell lines. And so we're going to use this to get 5 clusters with multitask graph clustering. And so these clusters are showing the similarity and differences across the 5 clusters. So essentially each one of these heat maps is 55 by 55, and the intensity of the red indicates how similar cluster 2 is, for example, in cell type I and cell type J. So you can actually compare them, and you can see that some of them look way more conserved and some of them look more diverged across the 55 cell lines. And so I'm going to focus on 2 examples, one of a very conserved cluster and one of a diverged cluster. And so here's an example of the conserved cluster. I'm only showing 6 different cell lines, but you can see that across the cell lines, there's parts of the network topology that are conserved across the cell lines, especially this bottom part right here. And so we can look to see what kind of pathways are there. And so if you look, for example, in fetal heart, you can see that there's a lot of terms related to ion homeostasis, which is important for cardiac action potential. But if you look across the 55 cell lines, you can see for the most part, these, and I don't intend for you to read this, but these are the ion terms, the ion homeostasis terms. You can see that it's actually enriched across the 55 cell lines as well. So while ion homeostasis is important for cardiac action potential, it's not unique to heart or fetal heart tissue. It's common in all cells and tissues. If we look at the cluster that looks more divergent across the different cell lines, so this green cluster here, we can see that it is changing a lot just in these 6 examples. Hopefully I didn't. Okay, there we go. And so we can see that this cluster is really lit up in fetal heart. And so if you look at the terms that are there, we can see that they're enriched for cardiac cell differentiation, for heart looping, basically things that are related to cardiac development. And if you look across the 55 cell lines, you could see, and again, these are the heart terms and the heart development terms. I don't expect you to read them. You can see that fetal heart is enriched, and there's not that much enrichment for these terms across the 55 different cell lines. So this is something that's more specific to fetal heart. It makes sense because it's fetal heart, and these are heart development terms. The ones that did have enrichment, or at least some enrichment, a lot of them were other fetal muscle tissue, which makes sense because they might share some similarities with fetal heart, as well as fetal stomach. So in summary, we developed an approach that can link regulatory SNPs to genes and look at the downstream pathways using graph diffusion, graph clustering, and multitask learning. The multitask learning really gives us a systematic way to compare across the different cell lines. And so I showed an application to the resting heart phenotype. And so we can look at conserved clusters and diverged clusters and see what different processes are there. And with that, I'd like to thank Shushmita and the rest of the lab, as well as Bill's group and Jen Sung's group, and our funding sources, as well as ISCB for the travel award. And if there's time, I'll take any questions.